Hello everyone, welcome back to my YouTube channel. So today we'll be discussing haematopoiesis. This is one of the core concepts for both physiology and pathology. So it basically all starts in third week of intrauterine life, wherein blood cell progenitors become the source of tissue macrophages and embryonic RBCs across many parts of the body. Like in brain you have microglial cells and liver you have the Kupffer cells. It started around 9 weeks of intrauterine life. Then, the mesoderm of the aorta or gonad or mesonephros gives rise to hematopoietic stem cells. And then later in the third month, these stem cells migrate to the liver and also a small contribution from the fetal placenta. So these cells can be useful for therapeutic hematopoietic stem cell transplantation. So now liver is the number one site of hematopoiesis till birth. And then fourth month of intrauterine life, the bone marrow develops, and after birth, this takes over the function of erythropoiesis. It's active throughout this entire skeleton, but puberty onwards, it's only the axial skeleton that's actively forming blood elements. So stem cells are pluripotent and have the capacity for self-renewal. In peripheral blood, these cells are mobilized during stress and on GCSF. We'll come to that later. So there's a multipotent progenitor which differentiates into a myeloid precursor and a lymphoid precursor. From lymphoid precursors, we get pro-NK cells, pro-B cells, and pro-T cells. NK cells are basically natural killer cells. So the differentiation of pro-B and T cells are called lymphopoiesis because they form pre-B and pre-T cells, which differentiate into B and T cells, which we know as lymphocytes. And of course, we have pre-NK and then the NK cells themselves. Myeloid precursors, on the other hand, differentiate into colony-forming units. One, they can be mixed CFU, and then you have CFU B or M or E. B stands for basophil, M stands for megakaryoblast, and E stands for erythroblast. They give rise to basophils, platelets, and RBCs respectively. Now, the mixed CFU differentiates into CFU G, M, and EO, myeloblast, monoblast, and eosinophiloblast, respectively. The myeloblast differentiates into promyelocyte, a myelocyte, then a metamyelocyte, and then finally band forms which form neutrophils. Myeloblasts can also transform into myelomonoblasts, which can form promonocytes, monocytes, and then macrophages. And then eosinophiloblasts, of course, give rise to eosinophils. Now let's look at erythropoiesis in detail. We have colony-forming unit erythrocyte, which differentiates into pronormoblast, which differentiates into an early normoblast, then an intermediate normoblast, then a late normoblast, which finally differentiates into reticulocytes, which give rise to mature erythrocytes. Now what you should know is hemoglobin synthesis starts in the intermediate normoblast, and the pronormoblast is basophilic with 2 to 3 nucleoli. The chromatin condenses in an early normoblast. The intermediate normoblast is acidophilic with basophilic hue, and the late normoblast has pycnotic nucleus and is acidophilic. Reticulocytes have no nuclei with RNA remnants, and the stain to visualize this best is brilliant crescile blue, which is also known as a supravital stain. Basically, stains that stain living tissue that has been removed from the body. Now, moving on to factors affecting erythropoiesis, we have intrinsic factors and extrinsic factors. Intrinsic factors are present inside the body, and extrinsic are basically dietary sources. So, we have hormonal factors and other factors. Like hormonal, we have erythropoietin, testosterone, growth hormone, and thyroxine. Others, we have intrinsic factor of Castle, which is required for B12 absorption, and hemolysates, or basically hemoglobin degradation products like heme. On extrinsic factors, we have B12, folate, iron, which is the main factor, proteins, and copper. Hypoxia increases erythropoietin production at the kidney. Something to know. Now let's look at the various colony stimulating factors or CSFs of hematology that influence erythropoiesis. MCSF stands for monocyte colony stimulating factor, and G stands for granulocyte. So these are the various sites in the erythropoietic situation where they influence the process. And interleukins influence lymphopoiesis as well at all levels, unlike GM-CSF, GCSF, and MCSF, it's just one stage. We also have cytokines influencing various stages as well. I will depict it here. Also, by the way, CFU-MG is also referred to as MK in a few places, so do not get confused. So, myeloid precursors also known as CFU-GEMM. So, G is granulocyte, E is erythrocyte, M is monocyte, and the other M is megakaryocyte. So, if you want to know some more in detail about erythropoiesis, something called a BFU or burst forming unit precedes the CFU-E stage of RBCs. So the other name for intermediate normoblasts is polychromatophilic. Late normoblasts are known as orthochromic, and early normoblasts is also known as basophilic normoblasts. So like any other hormone, erythropoietin works under negative feedback. It increases globin chain synthesis, enhances the maturation of RBCs, and release of RBCs into circulation. So how does it increase globin synthesis? It increases the delta aminolevulinic acid synthase activity. B12 is required for the maturation of nucleus and DNA synthesis along with folate as well. They have the same function. Coming to lymphopoiesis, we have primary lymphoid tissue which consists of the bone marrow, thymus. They're responsible for the proliferation of naive lymphocytes, basically lymphocytes that aren't yet antigen synthesized. And secondary lymphoid tissue is responsible for the proliferation of antigen sensitized lymphocytes. So where do you find it? You find it in the spleen, lymph nodes, and mucosal associated lymphoid tissue. So what are the stages of WBC maturation? You have a lymphoblast, then a prolymphocyte, and then a lymphocyte. Until this stage, it's all in the bone marrow. A lymphocyte maturing in the thymus under the influence of thymosin gives rise to T cells, and in the bone marrow you get B cells. Why B? The bursa of Fabricius in birds. I shall briefly depict diagrammatically B cell and T cell maturation. I recommend you read any microbiology textbook for the same because that describes this in detail. So a naive B cell migrates from the bone marrow to the mantle zone of a lymph node, wherein it migrates to the germinal center, and from the germinal center you can migrate to the marginal zone as well, where you get these various types of B cells. Germinal center is where the B cells actively proliferate. Now coming to the T cells, you have double negative T cells which after exposure become double positive T cells that express CD4 and CD8 on their cell membrane. So they then differentiate into single positives, that is CD4 T cells or helper cells and CD8 T cells or killer T cells. Now moving on to cell markers. So for RBC markers, we have hemoglobin, folate, B12, iron, ferritin, transferrin, and also erythropoietin, I forgot. For WBCs, we have CD45 as the pan WBC marker. This you have to remember no matter what. Specifically for T cells, we have CD4 and CD8, of course. CD1 represents thymocytes, and CD3 and 5 can be found in mature T cells as well, along with CD28. Also, germinal center cells and marginal zone B cells can give rise to plasma cells as well. We then have B cells, in which CD10 represents pre-B cells and the germinal center B cells. CD19 represents pre-B and mature B cells except the plasma cells. All cells expressing CD19 go on to express CD20 later on as they mature. CD21 is seen in mature and follicular dendritic cells. 23 is found only in mature cells, and 79A is found in pre-B and all mature cells including plasma cells. Monocytes express CD14, granulocytes express CD15, and before that, the Epstein-Barr virus receptor CD21 is highly overexpressed in Burkitt's lymphoma. CD11C is seen in monocytes, granulocytes, and Reed-Sternberg cells seen in Hodgkin's lymphoma. CD64 is expressed by all mature myeloid cells. Now, NK cells express CD16 and CD56. Myeloid progenitors express CD33. HSCs CD34, and CD30 is an activation marker for B cells, T cells, monocytes, and Reed-Sternberg cells. Now, for platelets, we have P-selectin, glycoprotein 2B, 3A, CD41, CD42, and CD62 as well. And P-selectin is also known as CD62P. Now let's see platelet production. How does that happen? We have megakaryoplasts, which mature into promegakaryocytes and then megakaryocytes. So megakaryoplasts undergo a phenomenon called as endo-reduplication, which is basically karyokinesis without cytokinesis, to form promegakaryocytes, which are basically like 32 times the diploidy of the megakaryoblast. They are highly basophilic with many granules. The megakaryocyte has a single multilobe nucleus with red-purple granules, and over 10 days it forms platelets. You can get 4,000 platelets from one megakaryocyte, which basically form as pseudopodia from the megakaryocyte. This whole process is influenced by thrombopoietin and megakaryocyte colony stimulating activity, also known as MEG-CSA. Okay, so small mistake here. These are all RBC biochemistry markers. So the proper red blood cell markers are A, B, and RHD antigens. For platelets, of course, don't forget we have CD41, 42, and 62. CD71 for RBCs and CD64 for monocytes. And of course, for immature granulocytes and monocytes, we have CD13. And for mature T cells, we also have CD28. So you can remember it like this: 1 to 8, 28 for T cells. For B cells, you can remember it like this: it's just 10, 19 to 23, 40, 79A and 79B, except 22, and leave out CD2, CD6, CD7 for T cells. So that's all for today, guys. Hope this was slightly helpful for getting these core physiology and pathology concepts clear, and I hope it helps for your exam preparation. So stay tuned for more. Like, share, and subscribe. If you have any suggestions, you can please comment down below. Thank you very much once again.